Hi, everybody. It's good to be back with you. I brought a friend this time because today we're talking about a changing virus, COVID, and how mutations, which now you've learned a lot more about those, but how mutations make a moving target for us as scientists as we try to address this pandemic issue. So I've got a friend, Dr. Mihail. Welcome, Dr. Mihail.
Thank you.
And we are going to go ahead and get this started. So, Dr. Mihail, do you want to start by telling us about the London patient?
Absolutely. So, the London patient is a man living in London who became sick with COVID, and he went to the hospital. And what was unusual about him was that he stayed sick with the virus persisting in his body for a whole lot of months, like 5 or 6 months, before unfortunately he finally died. And the scientists were interested in him, and so they took blood samples nearly every day. And when they did, they'd take the virus out of it, and they determined the sequence of nucleotides, so the genetic sequence of the virus.
We know about those.
And the thing that was surprising to them was every single time they sampled his blood, the virus was different. It kept changing. It was mutating.
Wait, so does that have something to do with like the variants we've heard about?
It does. It has a great
Okay,
deal to experience.
so we'll
And
get to that. But maybe first we should just do a little bit of background information about what a virus is really made of. Is that all right?
like a good idea.
Okay, so anatomy of a virus, guys. Basically, obviously, this is a cartoon, spoiler alert, but what you see on the inside is that the genetic information of a virus is RNA instead of DNA. And you guys have heard about RNA because it's involved in the gene expression that we're learning about right now. But my genetic information and yours and Dr. Mihail's, that's DNA. And so a virus is a little different from us. It has this single helix of RNA that makes up its genetic information. Other things you see in this picture, on the outside, all these fun spiky things, these are different kinds of proteins that have different roles in allowing a virus to be infectious and to attach onto a host. And we'll talk a little bit more about those proteins on the outside later. But the other thing I want you to notice is that there's not a lot going on the inside of the virus. There aren't a lot of organelles or parts of the cell, which helps explain why a virus can't replicate itself. It can't reproduce by itself. And that is why viruses need to have a host like us. And basically, these virus particles attach onto host cells, hijack their machinery or their organelles, and that's how they reproduce. So key things, RNA is the genetic information, got some important proteins on the outside, and because of the lack of other parts, viruses depend on hosts. Does that seem like enough information to get us started, Dr. Mihail?
I think so. I think that's a good introduction. Let's keep going.
Okay, so back to the London guy.
Okay, so the thing that the scientists found, as we said, is that all the time that this guy was in the hospital and they were looking at his blood, the virus kept changing inside his body. And one of the things they found, which really startled them, was there was one variant that was identical to a variant that had been found from a patient in South Africa and from a patient in Brazil. And
Wait,
so
so did the variant escape from him and go to those other places, or?
the big question, is it coincidence? Did the same mutations happen in different places? Or did that virus inside the London patient get out somehow? And that was what they were really concerned about, because if it was escaping and getting around the world, that's a problem.
Agreed. And you said there was another part,
There's another,
a complication.
so the London patient already had another disease, when he went to the hospital, he was someone who had what we call an autoimmune disease. And maybe you've heard of something like this hand. It's rheumatoid arthritis. It's an autoimmune disease of the joints. And so we should probably take a detour and think a little bit about autoimmune diseases.
Sure, yeah. I think this diagram might be helpful.
Ooh, great.
So what I understand about autoimmune diseases is that essentially you have the immune system of your own body attacking some of your own cells. Um, and that can cause all types of different problems. Am I on the right track, Dr. Mihail?
Yeah, that's exactly right. That's exactly right.
So
So
this would be a diagram for rheumatoid arthritis where the white blood cells, which are a part of your immune system, are attacking these cells in joints. And that's why, if you know anything about arthritis, you might hear about people having pain in movement because the cells that sort of cushion their joints are attacked. And they don't have as much cushioning, and there's some rubbing of bones on bones. Is that kind of on the right track?
that's exactly right. And the way doctors treat autoimmune diseases like rheumatoid arthritis is they give a medicine, a drug that makes our own immune system shut down. And so it helps the patient stop attacking their own joints, but it also makes the patient really susceptible to all other diseases because their immune system is being partly or completely shut down. So that's why for poor London guy, he was able to have the COVID virus in his body for months and months. His immune system was shut down.
Okay.
So
I have a question actually that I just thought of, Dr. Mihail. Are these treatments, so we said that our London patient has an autoimmune disease and his treatment shuts down his immune system. Are those treatments, do you know, are they specific to a part of his immune system that's, say, attacking his joint cells? Or are they shutting down larger parts of his immune system that are making it so that he can't defend against anything, like maybe COVID?
fortunately, it's the second scenario that you just said. The treatments shut down nearly all of the immune system. So people who take these kinds of treatments are now susceptible to things that most of us would fight off really easily. A cold can become a really huge problem for someone taking these drugs.
Okay. So then that's plenty of time, right, for the virus to mutate. Because if it's just replicating and you have some errors by chance, then if it's living in his body for months, more time for mutations, right?
New variants. And then what the scientists found out when they studied some of these variants from the London patient is they're easier, these new variants of COVID-19 virus move more easily from one person to another. So we say that they're easier to transmit, they move more easily. And they were very worried about that. And some of the other mutations make it easier for the virus to actually attack our cells. So those variants become more infectious. So
So
the two,
I remember a few months ago hearing about like the UK variant. I don't know if you guys, students, heard about that in the news, but I remember reading about that as one of the first like really scary variants where scientists were saying it's more virulent or like it's a bigger problem. Did that come from this guy?
that's the big question. Did it come from this guy? And we really don't have a definitive answer yet. This is a really good picture to help us kind of think about this question. So in this map, we have a lot of pretty colors, but the point is each different color is a different important variant. And if we look at the big green circle up near the top, that's sitting right on top of the United Kingdom, and the size of the circle says how many people we've found with the UK variant, and the green lines tell us where the variant has gone around different countries. And we can see it moving to Southern Europe. There's a big circle down there, and then some green lines over to the United States, green lines down to parts of Africa, green lines over to South America. So it's spread, and you can see that there's other variants, other colors that have moved as well. And so that's a big concern for trying to get the whole virus under control all over the planet. So did the UK variant come from this guy? Good question.
Okay.
And to get at that question, we need to think about another patient, another guy.
Yes, another story.
Now, so yeah, another story. So this is about a guy from Boston, the Boston patient. And he's a 45-year-old man, and he has another autoimmune disease. And so now we realize that he's taking drugs that are suppressing his immune system too. And that's a problem. So what happened with the Boston patient is he was taking his autoimmune drugs, and he developed a mild case of COVID-19. He went to the hospital. And while he was in the hospital, again, the doctors took blood samples, and the scientists took out the RNA from the blood samples, took out the virus, took out the RNA, determined the genetic code. And they discovered again that there were mutations because this guy had the virus in his body for months and months. The thing that made this an even more startling story was that the doctors in this case were trying to give the Boston patient some new therapy drugs to control the COVID. And when they did, it made the COVID virus mutate even faster than it did in the London patient.
Okay, wait, I have a couple questions now for sure. So, first one is, was this new drug to treat COVID, was that was he part of a clinical trial? Because we did briefly talk about some clinical trials recently in terms of ethics of vaccine development and things like that. But was he like a part of a trial? Or like, how did they not know that this drug had that effect? Do you think it was like in combination with his autoimmune disease?
I think that he was not, as far as I'm aware, he was not part of a trial, but I think there has been hope in the medical community that this particular group of drugs, which are supposed to help the immune system, would help this guy. But not only did they fail to help this patient, they, for some reason, gave the virus an even better chance to mutate.
Okay, so now I've got another question, because Dr. Mihail, like yesterday, we were learning about types of mutations, like how some mutations, if they're like silent mutations, guys remember those? But some mutations don't affect proteins at all, because you can change a nucleotide, but still have the same amino acid in a silent mutation. And so, like, did all of these mutations really matter, or were some of them silent mutations? Do you know what kind of mutations they were?
Actually, we do, and I've got a picture here to help us understand this. This is a beautiful picture, I think.
I like this picture.
So let's kind of take it apart. If we look at the left side, we see the COVID-19 virus, and Ms. Brun talked about just a minute ago, she talked about how the virus has proteins on the outside, and the red proteins are the ones we want to think about. They're called spike proteins, okay? So the little red triangle-y things on the surface of the virus are the spike proteins.
And this is a zoomed in spike protein?
I'm sorry?
And this is the spike protein, like zoomed in?
Here in the middle of the picture, we've got an actual picture of the way the spike protein looks. And you guys know by now.
This is a picture. This isn't just, you didn't just draw a bunch of spirals
No,
at home for fun?
a bunch of spirals.
Okay.
So the protein is the pink part and the red part. And you guys know about how proteins are made of a string or a series of amino acids, bound together. And you talked about how that worked, I'm sure. And then you found out that proteins have to fold in a really specific way in order to function. So if we think about this protein, the pink part up at the top is called the binding part. And then the blue protein is one of our proteins, and it's called
So
ACE.
that's not part of the virus. This is where our protein connects to the virus.
Exactly, that's exactly right. Our protein, the ACE2, connects to the spike protein at the binding place, the pink part. And it really, really, really matters exactly how the two proteins are folded so that they can bind together tightly. And so to answer the first question you asked then, in the London patient and the Boston patient, scientists found substitution mutations. And you remember those where there's one nucleotide that's swapped, that's substituted.
So
And
like guys, we exchange an A for a T or a C for an A. And then that, if that actually affects the amino acid, remember that you get from your codon wheel, then that's a substitution mutation. So I guess we found one of those that made it easier for the virus to attach to these proteins on our cells.
some of the variants that came out of the bodies of these two patients, there were several substitution mutations in this pink binding area that made it much, much easier for the COVID virus to bind to our protein, the ACE2 protein. And so that makes it easier for those viruses to infect us. And that means the virus, there needs to be fewer particles of the virus in our body in order to make us sick. And so that was really concerning, but that wasn't the only kind of mutation. So that's kind of that yellow box area where substitution mutations make it easier for the virus to bind. And if we look at the other end of the virus, down in the little blue-green box where the word spike is, there were deletion mutations there. And I think you guys heard about those too in class.
And we learned about how in deletion mutations, if you take away nucleotides or add them in some number that's not 3, right? Because there's 3 nucleotides in a codon, then that can really mess with a protein because the whole reading frame shifts and then every amino acid after it is affected, right?
Right, exactly. And so these deletion mutations at that other end, the spiky end or the green boxes, that part of the spike protein is what our body, our immune system focuses on in order to recognize the virus and fight it. And
So like our antibodies, because we've
our
heard about antibody testing and we know antibodies are proteins in our immune system that recognize like foreign things.
right, exactly. And so, for example, when somebody gets a vaccine for COVID, the vaccine encourages our own bodies to make antibodies to recognize the virus. Well, these deletion mutations mean that our antibodies either can't see the virus at all or can't see it very well. And so they're not as effective in fighting. So if a variant has either one of these mutations, that's a problem. If a variant has both of these mutations at once, that's really a problem.
Yeah, I can see why scientists would be worried about this and why then like vaccinations are so much bigger of a concern.
Absolutely. So right now, there are some questions that need to be answered in figuring out what we do having this information. Should scientists ask themselves, well, should we try and find people taking these immune suppressing drugs and make sure they all get vaccinated, even though we're not absolutely sure that these people are the source of new variants? Or should we wait and see if we can answer that question before we go in and vaccinate those people? Okay, so
So
that's
basically, should we change our whole like strategy, our whole timeline now, or kind of wait for more information?
exactly. Should we change now or should we wait? And that kind of brings a bigger question, I think, about how we make decisions about who gets vaccinated when.
Yeah.
That's a really huge issue right now.
Yeah, that's tough. And yeah, I mean, we talked about that in other areas of science ethics, just like who decides. It's kind of hard to wrap your head around that.
It
Um, I was kind of encouraging students, why not why not young people sometimes?
absolutely. And like if you look at what you're doing right here in Iowa right now.
Okay.
So here's your Iowa plan. I looked it up. And so you are now in Iowa in phase 1B of your vaccination program. In phase 1A, as I understand it, they vaccinated healthcare workers, which makes a great deal of sense. We need those people to be able to take care of us. And now you're in phase 1B. And some of the people in that phase are people like Ms. Brune, teachers
Yeah, that's
who,
why my arm hurts.
yeah,
Just got
exactly.
my shot on Friday.
So right now, Ms. Brune and teachers and other people who work in schools, first responders, people like firefighters and police officers and people who work with children. Then when those people are vaccinated, they move on to, it looks like they're going to move on to people who handle food, who process food, who perhaps people who work in grocery stores, that kind of thing. And so there's this whole plan for how you decide who's going to be vaccinated when. And if we think about the Boston patient, that's a 45-year-old guy who wasn't a first responder and isn't a healthcare worker and doesn't fit any of these early tiers. And yet we need to ask ourselves, well, this guy is sick and he's generating variants. And what if those can get out into the general population? Should we change? And I think Ms. Brune's question is absolutely right. We need to ask ourselves in a big picture sense, who should decide who gets vaccinated and in what order? Do we leave that to just doctors? Do we ask public health workers, people who study the spread of disease, epidemiologists? Do we ask government officials like the governor or the legislature, people like that? Should they have a say? What about ordinary people? What about, just people who are your neighbors or live down the street? What about you? I think that young people, personally, I think young people ought to have an active role in saying what they think, because the future is you. And it's important to know what you think and for you to express an opinion about who you think should make decisions, because until we control the virus everywhere, we're not done with this work. And that doesn't mean everywhere in Iowa, that means everywhere on the planet.
And if the why not you question sounds cheesy or like the future is you, um, we're in like real talk, right, Dr. Mihail? We're in uncharted territory. And what happens next, truth be told, is going to impact much larger of like your all's lifetime than it will of Dr. Mihail's. And so, I mean, there's something to be said for you guys, your vote should count for too. But and that's really where we're getting here. So first of all, Thank you, Dr. Mihail. I really think that we're all gonna just appreciate having honestly a real application of what we're learning about mutations, and I just really thank you for spending some time with us and for putting together these resources for us. So
Be happy to
thank
do
you.
it.
But Dr. Mihail posed a really good question here, and we're not going to stop there. So, should we modify the plan now, or do we wait for more information? Who decides? These are questions that I want you guys to spend the rest of your time here in class today responding to. So there is a discussion post. If you go to your Canvas calendar, I know you guys just love this thing. It's gorgeous, isn't it? It's probably your phone background. Anyway, Canvas calendar. There is a link here. It says COVID variants discussion post. And if you click here, I don't know how I can make that more obvious. Where you're going? Not clicking over here? No, just right there. Then you can respond to the discussion post prompt, which is what Dr. Mihail was asking. And you can also write questions for her. So like, she's like a human, like not even this like Zoom robot. And she is at home and would love to hear from you guys, any questions you have. And so when you're done responding to the questions she asked, ask her a question, and I'll put those together, send them to her in an email and get back to you with what she says. So also guys, I miss you still. But yeah, we're going to end it there. I'm going to give you some time to write. And I hope that today was something that you enjoyed and feel like you learned something from. So goodbye, Dr. Mihail. Goodbye, my wonderful people. I'll see you tomorrow with any luck. Bye, guys.